research on forms of vitamin E and their impact on allergic disease. Allergic asthma is prevalent in children around the world. At age 13 to 14 years old, 10% of the children in the United States have allergic asthma, and as well as some European countries. Other European countries are at 5 to 10% asthma. Allergic asthma occurs in about 80% of children with asthma and 50% of adults with asthma. Moreover, offspring of allergic mothers have an increased responsiveness to allergen, so there's an impact of maternal pregnancy and vitamin E isoforms during pregnancy and nursing on development of allergy in offspring. We've studied alpha-tocopherol and gamma-tocopherol forms of vitamin E and their impact on allergic lung inflammation, and shown that alpha-tocopherol inhibits, whereas the other isoform, gamma-tocopherol, even though it's at lower concentrations, is very potent and increases allergic inflammation. We've studied this in vitro, in animal models, and several clinical cohorts that I'll present today. There are several forms of tocopherols. There's 32 forms of vitamin E of tocopherol isoforms, including all the synthetics because of the racemic forms. The natural forms are alpha, beta, gamma, and delta, and there's 4 of these. Alpha-tocopherol is the highest concentration in your plasma. We've shown that it's anti-inflammatory, and gamma-tocopherol, which is about 10-fold lower than alpha-tocopherol, is much more potent than alpha-tocopherol and is pro-inflammatory. So those are the 2 isoforms I will be focusing on. Looking at the literature in the 2000s, alpha-tocopherol is approximately the same concentration around the world. It's between 19 and 25 micromolar most of the on the average. However, gamma-tocopherol varies among the countries. It's highest in the United States. It's about 5 to 7-fold higher than most of the other countries. Also, the Netherlands is about twice many of the other countries. Now, gamma-tocopherol levels in the plasma are determined by consumption. Alpha-tocopherol is not only consumption, but it's regulated by an alpha-tocopherol transfer protein that it's in the liver, and so that's why it's much more stable for alpha-tocopherol. The sources of the tocopherols are plants, and therefore the dietary oils, cooking oils. In the United States, we abundantly use soybean oil, corn oil, and canola oil, which is very rich, the red bars, in gamma-tocopherol. In contrast, sunflower and safflower oil, which is commonly used in Scandinavian countries, is very rich in alpha-tocopherol, which is the green bars. Mediterranean countries use olive oil, and that is mostly alpha-tocopherol, very little to no gamma-tocopherol. It's also been shown that the prevalence of asthma is highest in those countries that have the highest levels of gamma-tocopherol. So allergic asthma occurs when allergens are inhaled and impact the epithelium of our airway and therefore stimulate immune cells, dendritic cells, as well as these eosinophils that can degranulate and mediate the responses to allergens, including itch, airway remodeling, and bronchoconstriction. So the immune cells that respond are eosinophils. There are dendritic cells in this tissue, and I don't mean neuronal dendritic cells. Immune cells that are called dendritic cells respond and recognize the antigen and are important for initiation of disease. And my end of my talk is going to be on dendritic cells. There are also mast cells and lymphocytes that respond to the stimuli from allergens, and then fibrosis can occur through proliferation, and bronchoconstriction through stimulation of airway smooth muscle cells. In our animal models, we've used 2 different types of allergens: chicken egg ovalbumin inhalation, as well as house dust mite inhalation. After allergen challenge of these mice, we look at the airway inflammation, and eosinophils are a hallmark of airway inflammation. They look like this when they're stained with eosin. They have granules and a multi-lobed nuclei. We also examine the adhesion molecules on the endothelium because those are important for recruitment of the eosinophils, as well as cytokines and chemokines that signal for the eosinophils to come into the tissue. allergen-specific IgE, which recognizes the allergen, as well as the impact on lung function. In this first model I'm going to show you, we actually gave this tocopherol subcutaneously in a polyethylaxylated castor oil. This is the vehicle because tocopherols are in oil. And this is a vehicle used for clinically for oil, lipid, pharmaceutical compounds. And we wanted to do this because dietary changes in plasma tocopherol levels plateau after about 2 weeks on diet, and we wanted to be able to change this in 3 days, and it's been shown that subcutaneous injections can change to a plateau level in 3 days. So we allergen sensitize the mice with the allergen here. This is ovalbumin. We supplement with the tocopherol and then do an allergen challenge phase to look at the impact. And this does not have any effect on lung weight or body weight after these allergen challenges or after tocopherol supplement. So in this model, we measured the tocopherol levels in the lung and the plasma by HPLC with electrochemical detector. We measured alpha, gamma, and delta tocopherol. This is the basal level without supplementation. When given alpha-tocopherol in either the non-allergic or the allergic mice, this increased alpha-tocopherol in the lung and in the plasma. When given gamma-tocopherol, this increases gamma-tocopherol in the lung and in the plasma. And when given alpha plus gamma-tocopherol, it elevates both alpha and gamma-tocopherol in the lung and the plasma. Looking at the impact on allergic inflammation in the lung in these mice that had supplementation, the non-allergic mice are these first 4 bars that you can't really see. Allergen challenge increases the numbers of eosinophils in the lung lavage from mice that we've euthanized and collected the lung fluid. Then alpha-tocopherol reduces this allergic inflammation in the lung, the eosinophilia. Gamma-tocopherol elevates this, and alpha plus gamma-tocopherol has an intermediate phenotype not different than the controls. So this has an impact in interpretations. If plasma tocopherol levels are high in a human for gamma-tocopherol, adding alpha-tocopherol may not have as much effect because gamma-tocopherol is very potent, and I'll show you that in the next few slides. When we looked at lung function in these mice, this is a measure of lung airway hyperresponsiveness when the lungs are challenged with methylcholine. All the saline groups are here in black as expected. Allergen increases this airway responsiveness. Then alpha-tocopherol brings this back down. This is the blue one. Brings it back down to saline levels, so that reduces airway hyperresponsiveness. Gamma-tocopherol elevates this above that of allergic mice. So allergic mice with gamma-tocopherol have highly elevated hyperresponsiveness of the lungs. And then alpha plus gamma-tocopherol is almost like they were not treated. So these are mice that are allergic with alpha plus gamma-tocopherol, and they're the same as allergic mice that did not receive any of the tocopherols. Looking at the lung tissues, the brown dots here are the eosinophils. They're labeled for major basic protein. The allergic mice have increased numbers of eosinophils in the lung. Alpha-tocopherol dramatically reduced this. Gamma-tocopherol elevated the number of eosinophils, and alpha plus gamma-tocopherol looks very similar to the vehicle of the allergic mice. Saline mice don't have eosinophils. And if we quantify this, this is the allergic mice. Alpha-tocopherol reduces this, gamma-tocopherol elevates, and then this is the alpha plus gamma-tocopherol. When we look at the blood eosinophils, so these are the eosinophils in the blood ready to go into the airway spaces upon a signal for recruitment. And there's no change by allergen or tocopherol supplementation on blood eosinophils. So the source of blood eosinophils for the lung is about the same. When we look at the allergen-specific IgE, allergen increases allergen-specific IgE as expected, but alpha and gamma-tocopherol do not change this. So then we looked at another allergen called house dust mite and challenged the airway. And the mice were supplemented with diets this time. This is the standard diet. We used corn oil instead of soybean oil in our standard diet because soybean oil has very high levels of gamma-tocopherol. So we wanted to eliminate that difficulty. And more like 30 years ago, This was more of the standard mouse chow diet that used corn oil. Today, most of our standard mouse chow diet uses soybean oil. We supplemented this with alpha-tocopherol, gamma-tocopherol, one or the other. And then the basal diet has a level of alpha-tocopherol, which is 45 milligrams per kilogram, and that is very important because placenta development and therefore offspring require alpha-tocopherol. So you'll see there's basal, which means 45 milligrams per kilogram, 250, which means 250 milligrams per kilogram of alpha-tocopherol, and this one's gamma-tocopherol. So the mice were challenged with house dust mite. We looked at the lung lavage for eosinophils. There's an increase of eosinophils in mice on a basal diet that are allergen challenged, and then alpha-tocopherol reduces this. In contrast, we gave fewer house dust mite challenges to these mice in order to see the increase of gamma-tocopherol, because here is the maximum amount of eosinophils you really get in this model. So we, instead of doing 8 challenges, did 6 challenges. There is eosinophilia, but you can see gamma-tocopherol plus the allergen highly elevated the amount of eosinophils that went into the lung airway spaces. So I'm going to summarize the rest of the data on these experiments. There was alpha-tocopherol inhibition and gamma-tocopherol increase in tissue eosinophils as well as airway hyperresponsiveness. There was no change by alpha or gamma-tocopherol in blood eosinophils, ovospecific IgE, Th1 and Th2 cytokines for exposure to either allergen, as well as chemokine. This attracts the cells into the tissue, and there was no effect on VCAM expression by the tocopherol isoforms. VCAM is an adhesion molecule, vascular cell adhesion molecule 1 on the endothelial cell surface that is important for the eosinophils to bind to, and then eotaxin recruits the eosinophils across the endothelium and into the tissue. So here is a depiction of that. You have allergen challenges here. There's signals that activate this endothelium to express VCAM, and there's chemokines produced to attract the eosinophils across that endothelium into the tissue in order to respond and mediate the hallmark of allergic disease, which is eosinophilia. So we began to look at the recruitment process and regulation by tocopherol isoforms. My laboratory had already determined the VCAM signal transduction pathway through all of these signals in vitro and in vivo. So there is production of reactive oxygen species. And then this oxidatively activates protein kinase C. Downstream is protein tyrosine phosphatase 1B, ERK1, ZO1, and loosening of the junctions so that the eosinophil can roll and crawl in between the endothelial cells and into the tissues, called transendothelial migration. There's also an ICAM signal transduction pathway for recruitment of other leukocytes that also signals through protein kinase C. And this protein kinase C I'm going to show you, is regulated by the tocopherol isoforms. So we did this transendothelial migration assay where we grew the endothelial cells in a monolayer, placed eosinophils or leukocytes in general on the surface, looked at the number that bound, which is here, cell association, or the number that migrated between the endothelial cells, across the endothelial monolayer, underneath the endothelial cells. If we pre-treated the leukocytes with the tocopherol isoforms, there was no effect on adhesion to the endothelium or migration through the endothelial monolayer. If we pre-treated the endothelial cells with the tocopherol isoforms, there was no effect on the binding to the endothelium. However, there was an effect on migration after binding through the monolayer and underneath the monolayer of endothelial cells, called transendothelial migration, such that alpha-tocopherol reduced this migration, gamma-tocopherol increased it, and alpha plus gamma-tocopherol was very similar to the non-treated. So the tocopherol isoforms are affecting the endothelium during the migration assay that impact the recruitment of leukocytes and eosinophils into the tissue. So since VCAM signals through protein kinase C, and this is the structure of diacylglycerol that binds to the regulatory domain of protein kinase C, and it has a similar structure to the tocopherol isoforms. There's a large head group here with a lipid tail. We determined whether these tocopherols bound to this regulatory domain and could compete with diacylglycerol. So this is an assay where the tocopherol is fluorescent, and that's the background fluorescence by the concentrations of labeled MBD labeled alpha-tocopherol. When it binds to a lipid pocket on a protein, it increases fluorescence. And so alpha-tocopherol, when we gave it the C1A domain of protein kinase C, it increased the fluorescence. So here's the background fluorescence of the tocopherol. It's increased when alpha-tocopherol binds to that lipid pocket of protein kinase C. And then unlabeled alpha-tocopherol competes for binding, so it's a specific binding to the C1A domain of alpha-tocopherol. Interestingly, it took about 50 micromolar to block the labeled alpha-tocopherol binding to that protein kinase C. In contrast, only 10 micromolar is needed to compete for the labeled alpha-tocopherol. So gamma-tocopherol outcompetes alpha-tocopherol for binding to protein kinase C. Also, alpha-tocopherol competes for diacylglycerol, this is dioctanyl glycerol, for binding to the protein kinase C regulatory domain. And a control lipid cholesterol does not compete. When we looked at the activity of the enzyme, this is purified enzyme, alpha-tocopherol blocks that activity, gamma-tocopherol increases it. When we analyze protein kinase C activity within the endothelial cells, so this is not purified protein, this is PKC within endothelial cells stimulated by anti-VCAM, which cross-links VCAM and activates PKC. This activates the autophosphorylation of protein kinase C. The white bars are the non-stimulated anti-VCAM cross-linking increases activation of PKC. Gamma-tocopherol elevates this. Alpha-tocopherol blocks this activation of PKC. Alpha plus gamma-tocopherol has an intermediate phenotype that's similar to the control. So, in summary, the alpha-tocopherol binds to protein kinase C, so does gamma-tocopherol. Gamma-tocopherol is more potent for binding and outcompetes alpha-tocopherol at that site. And in vivo and in vitro, gamma-tocopherol can block the anti-inflammatory effects of alpha-tocopherol. So gamma-tocopherol is pro-inflammatory, outcompetes alpha-tocopherol, and alpha-tocopherol has anti-inflammatory properties for allergic disease. So we looked at this in humans. This is the Shanghai Women's Study, and this is low, medium, or high levels of tocopherol with increasing concentrations of alpha-tocopherol. And if we just focus here, this is high gamma-tocopherol with low alpha-tocopherol. And in that circumstance, there is 3.5-fold higher odds ratio for developing allergic asthma within 8 years. In another study with Cardia, this is a U.S. study with multi-sites, 4,500 individuals. There are measurements of alpha and gamma tocopherol in blacks and whites in this cohort. When we look at lung function for alpha tocopherol association with forced expiratory volume, which is blowing out air within 1 second, and forced vital capacity is blowing all of your air out. So alpha tocopherol has a positive association. These are positive values. This has a trend for FEV1 and significant for FVC. Then gamma tocopherol has a negative association. These are negative values. So as gamma tocopherol concentrations increase, there's lower FEV1, FVC, which is worse lung function. And this is highly significant in blacks and whites. Then when we look at the tocopherol isoforms and we put them in different groups based on European and Asian levels, so the low 1 to 2 micromolar, some intermediate concentrations, and the greater than 10 micromolar. So the greater than 10 micromolar is highly elevated gamma tocopherol, has lower FEV1 by age 21. Lung function declines with age, but it also is much lower if there's high gamma tocopherol. This is true for all participants, non-asthmatics, as well as self-reported ever asthma. And then FVC has the same pattern. So this develops early in life and is established by age 20. So in this cohort, there was 500 mLs lower FEV1, and this leads to a 10 to 17% decrease in FEV1, which is forced expiratory volume in 1 second. This is actually a higher percent decrease than one would get from occupational allergen exposure, which is 5 to 8%, particulate matter, which is 2 to 6%, cold exercise, which is 5 to 11%, and dander, which is 2 to 8%. Based on this prevalence of 10 micromolar and the census in the U.S. in 2011, there would be 4.5 million people in the U.S. with 500 mLs lower lung function. Since this occurred by age 20, we looked at the development of allergy in neonatal mice from allergic mothers. This is relevant to prenatal vitamins as well as early life nutrition. When pups of allergic mothers are exposed to allergen, they do not develop allergy. Pups of allergic mothers given the same suboptimal allergen challenge develop allergy. If the mothers are supplemented with alpha-tocopherol, this blocks the development of allergy in the offspring. If the mothers are supplemented with gamma-tocopherol during pregnancy, this highly elevates allergic inflammation in the offspring. For this model, the mothers were supplemented with 250 milligram per kilogram of tocopherol, alpha or gamma. So the mothers were made allergic. Then at time of mating and during pregnancy and nursing, alpha and gamma tocopherol diets were supplemented. Then the pups after birth were allergen challenged and pup tissues assessed for allergic inflammation. So when alpha-tocopherol and gamma-tocopherol diets were supplemented, there was an increase from basal levels of tocopherol with the supplementation in both the mom liver and the pup liver. In the mom and pup, there is some decrease in alpha-tocopherol if the mother was allergic. This is consistent with what happens in humans. There's lower levels of alpha-tocopherol in allergic individuals than non-allergic individuals. When gamma-tocopherol is supplemented, this also increased gamma-tocopherol in the tissues, but if the mom was allergic, these levels were lower, suggesting that there may be an increased need for tocopherols during allergic disease. Also, alpha-tocopherol had a slight non-significant increase in percent of pregnancies as well as number of pups per litter. In contrast, gamma-tocopherol significantly reduced the number of pregnancies and had a trend towards about half as many pups per litter. When alpha-tocopherol was supplemented during mothers during pregnancy and nursing, this decreased the amount of eosinophils in the pups of allergic mothers. Gamma-tocopherol had the opposite effect. Pups of allergic moms had allergy, but if gamma-tocopherol moms are supplemented with gamma-tocopherol, this highly elevated the allergic inflammation in the pups. There's no effect in ova-specific IgE in the pups or the mothers with alpha or gamma-tocopherol. Alpha-tocopherol did reduce several of the cytokines and chemokines by both qPCR and protein, and gamma-tocopherol elevated this. So overall, in offspring of allergic mothers, alpha-tocopherol supplementation in the mom during pregnancy and nursing decreased the allergic inflammation in the offspring. Gamma-tocopherol increased this. I didn't show you the data, but it also affected the number of dendritic cells in the offspring, and those are the cells that are important in initiation of allergic disease and recognition of the allergen. This occurred in the fetal liver and the pup lungs, in vitro cultures with bone marrow cells, and in dendritic cell PKC alpha activation. So in summary, alpha-tocopherol inhibits allergic inflammation, gamma-tocopherol elevates allergic inflammation. This occurs in adult mice. There's an association of alpha-tocopherol with better lung function in humans and gamma-tocopherol lower in lung function in humans by age 21, so it develops probably during childhood. Also, alpha-tocopherol supplementation of allergic mothers blocks the development of allergy in offspring. Gamma-tocopherol supplementation of allergic mothers increases development of allergy in offspring. Gamma-tocopherol also increases the numbers of dendritic cells, a subset of dendritic cells that are important for initiation of allergic disease. And both in dendritic cells and in endothelium, alpha-tocopherol blocks PKC activation and gamma-tocopherol elevates PKC activation. So in general, Summary: alpha-tocopherol inhibits allergic inflammation and gamma-tocopherol blocks. And I want to thank the people that are in my research group. This is some of the current group, and we went to a baseball game and had a lot of fun. There are many collaborators around the world. This is our building. I would thank the National Institute of Health in the United States for funding and the Well Center for Pediatric Research. Thank you.